one that I thought would be relatively understandable given what we've talked about recently. So what we're going to do is we're going to do basically a jigsaw style for talking about this paper. The reason why I broke you into groups today is that there are 9 different prompted questions, all focusing on a different aspect of the paper. So for example, one of the groups has one that says, how did the authors decide on the experimental design for hand washing in the experiments described in figure 1 and 2 and 4? What led them to choose the time, temp, and percent triclosan that they did? What are the 2 categories of chemicals used in antibacterial soaps? So that is one of the prompts. And ultimately what I want you guys to do is I'm going to give you about 5 minutes or so to just answer these to the best of your ability. And then we'll talk through them. And your group will be the leader for that particular question. After we finish discussing this paper, I will collect all of these and then scan them so that you have them as notes as well. But what I want you to do is just start with some ideas, and then as we discuss, somebody be the note taker to continue adding things if there are things left unsaid. So modify. And this is where we'll start working on what's a good answer versus what's the best potential answer you have. And here, or in this particular paper, it's about the details. It's about the details, okay? So, with each of the groups, have somebody start taking notes, start work on that prompt, take your papers out, make sure that you have them available with you to be able to mark down some answers, and we'll reconvene in about, let's say about 5 or 6 minutes. Okay, we'll see how we're going at that point.
More effective at killing the bacteria as marcescens than regular soap.
Okay. So, regular soap or soap with triclosan, so an antibacterial, which one is more effective at killing bacteria during hand washing? Okay. And then what do you guys have for the other parts? What's the importance of the research question, and then what's the why was the FDA considering a ban?
So we put it is important because of the antibacterial soap is not more effective and it can have more negative effects like allergies, antibiotic resistance, endocrine disruption, acute and chronic toxicity, and bioaccumulation. And then those were the reasons that we had for the FDA considering a ban.
Okay. So basically, the question is it more effective? And if not, why continue to include it? If the drawbacks are worse than the benefits, right? Anybody want to add anything to that? The one thing that I might add, what is the function of hand washing? In terms of how we described in terms of describing it as a sterilizing, disinfecting, antiseptic, or degerming event, what is hand washing? Devin? It's degerming. Are we trying to kill bacteria per se in a degerming event? And the answer is no. By definition, it's mechanical removal. It doesn't define whether the microorganism is killed or not. So though the question may be whether does the antimicrobial soap kill, I absolutely believe that's why it was included in the first place. There's a disconnect here, right? One of the things that we talked about with antimicrobials is that it's not only the concentration that has an effect on growth of organisms, but what else? If concentration and something else is required for there to be an inhibition of growth. What was that? It is time of exposure. And in hand washing, what's the typical time of exposure?
Like 20 seconds.
20 seconds or less, right? I would bet on average if you were to go and pool 15, 20 people outside of this classroom, it's going to be less than 20 seconds. Is that enough time to kill? Probably not. And so there's a disconnect here. And this is really why the FDA started looking at this, was that the time of exposure really wasn't enough to have an effect on somebody's hands, at least in those first few minutes. Okay? Good. Anything else to add? All right. Let's move on to the second one. Who had this one? How did the authors decide on the experimental design? Okay, Ashley, go ahead.
So figures 1 and 2, they also the temperature between 22 and 40 degrees because those were the temperature recommendations for hand washing, and they used 20 seconds for the same reasoning. In the figure 4, they chose to measure the colonies on the 16 volunteers using the plain soap and antibacterial because the most antibacterial soaps have varying concentrations of bacteria.
Yeah. Where did the recommendations or those guidelines come from? I think they state a couple of sources, don't they? And anybody can answer this one if they know it. Let's take a look at the materials and methods real quick. So in the materials and methods, under experiment 1, the in vitro antimicrobial testing, second paragraph. Yeah, Healthcare Infection Control Practices Advisory Committee, the World Health Organization, the Centers for Disease Control, and the Ministry of Food and Drug Safety in Korea recommend that consumers wash their hands 20 seconds, the time it takes to sing Happy Birthday twice. So based on the recommendations from those 4 different entities, that's where they came up with the protocol that they're going to use in this particular study. I do remember they mentioned one other. American Society for Testing and Materials Standards suggests water temperature to be 40 degrees Celsius. So that is kind of a standardized way to test these antimicrobial soaps against one another or against just regular hand soap to determine its effectiveness. Okay, so to say, now, would I expect you to memorize particularly those groups on exam? No. But if you have this in front of you, that would be the detail that we're talking about. Not only saying what the recommendations are, but who did they come from? I mean, they could be from Uncle Jack's coffee and pastry shop on hand washing, or British boutiques and essentials. And I don't consider those either to be reputable sources on hand washing. So, whose recommendations are we referring to? All right. You guys have a drawing for 3, right? Abby, do you want to bring it up? Display it so we can all see it. So for this third one, what I asked them to do was to actually model this. Model how the experimental design was set up. That's okay. It doesn't have to be perfect. Lame is cool. Do you want to explain it as you're walking through it?
So we were asked to do a diagram of figure 1A and B, kind of like what the process they use. So these are the 3 test tubes, the control tube, the one with the 0.3% triclosan, and then the plain soap. And then these arrows are just showing they put the bacterial strains in, that they incubated them for 20 seconds at 22 degrees C. And then we didn't put on there, but they add like a agent to stop bacterial growth after that. And then serial dilution of those plate count, and then they incubated the plate count to allow the colonies to grow, and then they counted it.
And then what equation would they use to determine the original concentration? Anybody know? I know you know. Anybody remember the equation we used? CFU divided by what? Volume that was plated times the dilution. So that's how they would go back and determine these. I think this looks good. What's the modification difference in figure 1B? Or sorry, no, not 1B, 2. Temperature, right? So figure 2 has this temperature at 40 degrees Celsius for that period of time. Right? Okay. And then thank you. I will come and grab it later. One modification to point out is the stuff that they added to the soap afterwards wasn't actually to inhibit the microbial growth. It's actually to inhibit the antimicrobial effects. It's to neutralize the triclosan and the detergents that are in there so that they don't continue to inhibit microbial growth at that point, which is necessary because if your only way to count cells is to measure growth, you need to inactivate those disinfectants. Okay. Let's do one, actually, you guys had number 4, right? No? Who has number 4? Oh, I screwed up my order. Okay. What was the question for 4 again? Because I can't remember off the top of my head. Oh, perfect. That's what I was going to suggest that we do next. Okay. So, describe... Decrease in bacterial growth when comparing what? The soaps, right? I would argue, no, there's a decrease. Where would I be correct in saying, yeah, there's a decrease? From washing and non-washing. So that detail needs to be there to be able to make sure that we're identifying exactly what we're talking about. So you're absolutely right, though. Between soap with triclosan and soap without triclosan, there is literally no difference whatsoever. And I can remember reading somebody's response last night. I didn't get to all of them, but I read somebody's and they're like, but that's not supposed to happen. And my response was, it's not about what is supposed to happen. The results don't lie. It's our interpretation of these is what lies. Okay? And so we have to take the results for what they are at face value. And here we're finding 20 seconds of hand washing. Well, in this example, it's not really hand washing. It's in a test tube, right? 20 seconds of incubation between Enterococcus faecalis and soap with or without or the original population. And what do we find? What's the difference between without washing to washing with just regular soap?
It does decrease, but then you don't see the difference between
Right, there is a decrease, and if we could quantify that decrease, what is that amount of decrease? Anybody remember how to interpret a graph like this, where the y-axis is its bacterial growth, but what are the units? Log CFU per mil. The way to interpret this one, anybody know before I describe it? Sort of. This is an exponent. So, for example, all you have to do to convert this to something that might make more sense is just put a 10, and these numbers represent the exponent. In other words, 0 is really 1, or 10 to the 0. 10 to the 1 is what? 10, for all of you math geeks out there. 10 to the 2? 100. 10 to the 3? 1,000. 10 to the 4th? 10,000, yada, yada, yada. Okay? So in other words, 10 to the 6th is 1,000,000 cells. 1,000,000 colony-forming units of Enterococcus faecalis per milliliter of sample that they're testing. That drops down to 10 to the 5th, or 100,000. A couple of ways that we can describe that. We can say, 1, there's a 10-fold decrease. Or another way we can describe this is that there's a 90% reduction in the amount of bacteria in this sample just by incubating it with soap for 20 seconds. And this is important because in bacteria, a lot of times in our world we talk like, yeah, twofold differences are statistically significant. It means jack in the microbial world. If you ever look at a disinfectant, for example, like a Lysol wipe or Lysol Clorox bleach toilet cleaner, how effective are they? When they report percent effectiveness, what do they say? They are 99.99% effective, meaning if you have 10,000 bacterial cells, it is implicating itself saying that it kills all but 1. That's significant. But going from 100,000 down to 50,000 bacterial cells is not significant, typically. So when we're looking at bacterial populations, if we're trying to determine is there really an effect, we're looking at 10-fold or greater changes, 1 log changes, increase and decrease. I think the other point that was made, no real difference between gram-positive and gram-negatives. If we were to describe the general trend here, yes, there are outliers. For example, Streptococcus pyogenes really gets wiped out by hand washing, which is probably a good thing. That one causes, anybody know what Strep pyogenes causes? What did you think you had last week? Well, other than that, what is scratchy throat? Meaning, anybody had their tonsils removed? Were you guys exposed to a lot of strep throat in your time? Yeah. Strep pyogenes causes strep throat. Hey. All right. So it's a good thing that one's being killed. But most of these are maybe in that 20-second exposure, only decreasing by about 10-fold. And I think if we look at those gram-negatives, we see the same sort of effect. 10-fold, maybe even less. Maybe even a little bit less. Okay. Oh, we are out of time. So, before you all pack up, here's what we're going to do. I need to give a couple of notes for upcoming.